about COVID, and we will talk quickly, and I'll answer any questions you have about COVID. Why do I know about COVID? Because all the people like my age group who are infectious disease specialists were kind of forced by the NIH to be COVIDologists, and we became COVID leaders. So you kind of see and this is interesting because you see in the local campus certain people doing stuff. You see on the news, mostly people who aren't really working on COVID but want recognition. This is the national news. And then there's the actual people who are responsible for all the research work being done. So a lot of it's almost all being done with federal dollars, and most of those federal dollars are flowing through HHS, some of them into a thing we run called the COVID Prevention Network. So we're responsible for the development of vaccines and other preventions for COVID. And then another one of my colleagues is responsible more for treatments, although I'll talk about both. So this is more of a national level view of what we're doing, but that's okay. So shall we begin?
That sounds great.
Why don't you interrupt me since you're unmuted, or can you interrupt me if something doesn't make sense, you want to talk about it?
Okay.
So here's the problem. Coronaviruses, and they all look kind of the same, but they don't all use the same receptor. This virus is called the coronavirus cleverly because it's got this corona when you look at it on an electron microscope. And we know that for the SARS-CoV-2, which is the third of these terrible coronaviruses we've dealt with, it's SARS-1, MERS-SARS, and our SARS-CoV-2. We know that this is what the three-dimensional view of the spike protein looks like. And we know that the spike protein wants to find a receptor on a cell called an ACE2 receptor. They're expressed all over the body, but our problem with this virus has been the ACE2 receptors in the nose. The nose is very rich in ACE2 receptors, and interestingly, the nose has a lot of different parts, and the smelling part of the nose is particularly rich in ACE2 receptors. So the nose, infection of the nose, is the window to this infection. If we could prevent infection in the nose, you're not going to get SARS-CoV-2. And once it's in the nose, most people are going to do well anyhow and eliminate it through their own host defenses. And some people are going to go to sleep with SARS-CoV-2 in their nose, and when they sleep, they're going to aspirate the virus into their lungs, which happens all the time. Fluid falls into the lungs. And if you have bad luck, enough of this fluid falls in your lungs to bind ACE2 receptors in the lungs, you're going to get pneumonia and be very sick. And very sick is kind of shown on this pretty complicated slide. Worldwide, 100 million people have already gotten this infection. We don't really know how many people have died worldwide. We know the US, every state's been affected. We know we've suffered 2 waves of this infection. And we know we've got at least 20 some million cases, at least 450,000 deaths. So it's a bad disease. It's not that much fun for us as an epidemic, to be perfectly honest. So this is really important to understand everything you see on the news. No one ever talked about R0 or R sub-0 before a year ago. And now everybody wants to talk about this number. But this is the number that infectious disease specialists use to try and prevent all epidemics that involve humans for human-to-human transmission. This formula allows one human's risk to another human. It's first described by a guy named Roy Anderson, who's still very active. Bob May is dead. A long time ago. But for people like me who spent their career in this business, this formula has been critical. So we want the R0 to be less than 1, with the argument that epidemics implode if R0 is less than 1. So how do we calculate R0? We multiply beta, the efficiency of transmission of the pathogen, times D, the duration of infectiousness, times C, the number of people exposed. 3 metrics. So you can kind of see that one bug might be incredibly contagious compared to another bug. For example, HIV, which is sexually transmitted, is not very contagious. It takes a ton of episodes of intercourse, at least heterosexual intercourse, penile vaginal intercourse, for a transmission event to occur. Whereas respiratory viruses are much more efficient from going from one person to another. And again, the nose is the window to the problem. This guy, Sloan Fussell, President Fussell, as he wants to be called, President Fussell is covering his nose, which is a really desirable thing because that is where this virus is going to go to the next person. And then the next person standing near him, if they've covered their nose, you've drastically reduced the probability of a transmission event. We've reduced the efficiency of transmission in a mechanical way, in a behavioral way. Things that are more efficient are worse, things that are less efficient are better. Why are we so worried about these variants? Because the argument has become, for a variety of reasons, that the variants we're seeing in South Africa and Manaus, Brazil, that they're more transmissible. The virus has evolved so that it goes more easily from one person's nose to another person's nose. And the second thing is duration of infectiousness. The shorter the infection, the easier it is to deal with. So SARS-CoV-2 has a pretty short contagious period. You know, your nose becomes positive and you're not contagious forever, although the newer variants look like you're contagious longer, which makes the newer variants worse. But mostly after a few days, you stop having what we would call replication competent virus in your nose. And lastly, there's a number of people exposed. For STDs like HIV, hopefully you're only exposing one person at a time. But for a respiratory pathogen, somebody can sneeze while singing and infect a lot of people all at the same time. So we have two big disadvantages here. We have an advantage that duration of infection is thought to be short, but the number of people exposed can be very large, and this can become very, very efficient. Hence, we're sustaining a gigantic, terrible epidemic that's ruining your lives. And the spread of this disease is to some extent age dependent, not because one group is more susceptible, but because the behaviors of the group. Younger people see themselves as less vulnerable. And in fact, the data, as I'm going to show you, suggests you are less vulnerable. Not less vulnerable to being infected, but less vulnerable to dying from this infection. I am going to show you there's a very linear relationship between age and badness. So we see that young people get a lot of infections, and you could see we have these waves of infection in dormitories and on Franklin Street and so on and so forth. And then it gets better, and it gets bad again for reasons we don't completely understand at age 50 to 64. And it gets much better when people get older because they're so afraid of getting infected. So the infection rate goes down, but the consequences of infection are much more terrible. And this, by the way, just thrown in there is just to show you that every university, not just UNC, that's tried to bring students back has problems with the spread of virus. It's very difficult to live in a dorm situation and not get infected. It's difficult to have a social life and not get infected. I mean, it's obvious, you know, it's not rocket science. So you'd say, well, like, look at Duke, they did such a great job, you know, the first semester, fall semester, where we sent you guys home. Let's talk about that for a second. We sent the students home in the fall because we didn't, I guess it was, the fall. We didn't know about the morbidity of the infection. We were afraid that if too many students got infected, some of you would die. And that was totally unacceptable. So in 3 days, there were 1,000 infections on this campus. The decision was made to send the students home. Now we're much more sanguine because we don't think that, the infection will not have as bad consequences in younger people. So we're less afraid than we were in the fall. Now, Duke did all kinds of bells and whistles with measurement of COVID, which was good, but mostly they had success because they had kids living in one room, no Greek life, no upperclassmen, and they went from Whole Foods to their dorm room and back. You know, if that's what you do, there's a pretty good chance you're not going to get infected. So this semester now we're doing much better. Students are much more responsible. The living conditions are less crowded, and we're testing students much more aggressively than we were before. This is just an example of morbidity and race. And what's shown, you look at dark color is white, orange is black, and gray is Hispanic. And you look at the bars and you see that the ratio of the death rate. So somebody gets COVID, are they going to die? You see that the probability of badness happening is much higher in African Americans and Hispanics. Why is that? They have much less access to good health care. There's a higher infection rate, first of all. Poor access to good health care, delay in getting good health care, and a lot more comorbidities, a lot more obesity and other comorbidities because of lack of good health care and some poor nutritional. So we know, and this is very important to us in terms of dealing with our interventions, to not just deal with the kind of wealthier, better to do people, but to deal with the entire population of the country. So the disease itself, so now you've been exposed to SARS-CoV-2 and you're a young person. About half the people who are exposed never have symptoms. They remain asymptomatic, especially younger people. We know they become infected either because we sampled their nose and found the virus, or they briefly seroconverted. But asymptomatic infection is much less powerful in making you seroconvert and showing you were positive. So you don't get all the immune benefits of being symptomatic. Symptomatic can be mild illness, moderate illness, severe illness, or critical illness. Each has a definition because when we do experiments and clinical trials, we have to use these definitions to decide if what we did made any difference. So you can see mild is you just didn't feel well, almost anything. Moderate is you got to start having signs that your lower respiratory tract is being affected. Severe, you're in the hospital, you're really sick, and critical, you're in an ICU. Let me ask you a question. Is this the lecture you wanted to hear? Can you understand me at this pace?
Yeah, we can understand. Thanks.
Is this what you want to hear about COVID? We could spend the whole time answering questions about COVID, or I can take you through everything we've done. This is a democracy. How many of you are there?
I think it's good to go through everything. You know,
How many of you are there, Amber? You're going to be the spokesperson.
I haven't heard half of this stuff, so I think it's extremely interesting.
But how many of you total are there?
There's 22 of us.
Okay, 22. Anybody can stop and say, there's a veto power. Anybody can say, I am bored, stop and let's just answer questions. Okay.
Can I ask one quick question? Because you had touched on how asymptomatic individuals don't get the benefits of, I guess, like a immune system strength against COVID. Could you kind of like elaborate more on that?
Well, we're not really 100% sure of it. We know that if we take people who are asymptomatic and we look at their blood, in the peripheral blood, they lose the antibodies that would demonstrate infection 50% of the time. What we don't know is what happens to the cells in their body that might have a good memory. So we don't really have enough experience yet to be sure that asymptomatic infection isn't helpful, but it's probably certainly, we know for sure it's not as helpful as severe infection in terms of the immune response. And I'll show you that in a couple of minutes.
Okay, interesting. Thank you.
So this is kind of how we think about this. You know, you became infected, you're either asymptomatic or pre-symptomatic. If we sample your nose and it's positive, we don't know if you're going to go on to symptoms. But in young people, about half the people will remain asymptomatic. And then some people, and the virus will replicate to its highest concentration. Now it's going to get about, billion, 10 to the 7, 10 to the 8th copies in the nose. And that's really important to us. The nose copies matter. And most people are going to reduce the copies in the nose over the first week, and they're going to get better. And they might have lingering symptoms, but they're not going to die. But some people won't clear their nose, and they're going to get sicker, and they're going to get hospitalized, and some will get very severe illness. And then it's not the virus making them so sick, it's the immune response to the virus. And so we have all these cookbooks to try and prevent death and destruction. This is a really important slide for you to look at carefully. You are the comparison group. We use you as very unlikely to get very sick or to die. We say that a little kid, 5 to 17 or younger, they are even less likely to get sick and die. It might take 30,000, 40,000, 50,000 kids for one to get really sick. But look at what happens with age, hospitalization, 2 times greater, 3 times greater, 4. It's linear. It goes up and up. And then you're 75 years old, you got an 8 times greater chance than the young people on this call of actually being hospitalized. And you look at death, you look at age 75 to 84, you see a 220-fold greater chance of dying than a college student. So all of our energy, rightly or wrongly, is spent on trying to keep people, older people, usually we focus on people over 65, as much as we can from getting COVID because they don't do well. People 55 and older with comorbidities that include kidney disease, diabetes, obesity, especially obesity, they're also more likely to do poorly. So we have a cookbook of how to of who to look at to try and help people from getting sicker and dying. Now, we're going to come to, on this bottom of the slide, is something that's going to be emphasized over and over and over again. You don't need to get this infection. Most people can prevent getting infected through behavior changes that are risk reduction. And just like we ask you to have safer sex, we ask you to have safer respiratory status by wearing a mask and keeping apart, washing your hands. The hand washing and all that, we're not really sure how much that helps. The masks are what really matter. And if you're not wearing a mask, distance matters a lot. Just today, the CDC has made the argument that a lot of the masks that are being made are insufficient. And so we're going to try and ask students to wear a cheap paper mask and then a cloth mask with ear loop connectors, like all of you got the UNC mask that you can tighten. So tightening a cloth mask over a cheap paper mask actually will be a better preventive than wearing a paper mask or a cloth mask alone. This slide just emphasizes what I've already told you. The COVID, 5% of infections are in long-term care facilities, and 35 to 40% of all the deaths are in long-term care facilities. So this is where we are. Terrible pandemic, a lot of people infected. This is not acceptable. So for every infectious disease, we have 3 things to do to make R naught less than 1. The first thing is behavior change. Try and incite safer behaviors, which in this case is just wear a mask reliably. It's so easy and yet has been so difficult in the United States. But we know this works because we know China has simply aborted their epidemic by wearing masks. We know Vietnam, where masks are very reliable, has 100 cases of COVID. So the tragedy in Europe and the United States is palpable and terrible because we couldn't inspire behavior change. But beyond behavior change, then we need biological solutions. We in the West always gravitate to biological solutions. And we have a couple of biological solutions. One is let's make a vaccine that works and vaccinate people and hope that we can prevent infections and transmission and death with a vaccine. Another thing we can do is make antibodies in a lab very easily and infuse those antibodies, and that gives you instant passive immunity because we're basically doing about 2/3 of what a vaccine would do. And we can make treatments. So when somebody gets COVID and is a risk group, we can say, wait, you've got COVID. This is the 2nd day of your symptomatic illness. Come to CVS. They now have these little boxes outside CVS. Go in a little box and let us give you an infusion of a drug to try and prevent progression of disease. And that drug is, again, going to be an antibody to try and wipe the virus out of your nose. We're going to try and reduce the concentration of virus in your nose as fast as we can to protect your lungs. We suffered the great flu pandemic, and they had the same issues we have. They had to wear masks. There was an anti-mask society. They had like all the same problems we had for 2 years in 1918, 1919 flu pandemic. So the species is not new to the problem of inspiring behavior change. So biological strategies for prevention, treatment and prevention. So one of the biological strategies is going to be the treatment of an infection where treatment also serves as prevention. How does that work? If we have a drug to give you that stops you from progressing your disease, and the drug reduces the concentration of virus in your nose and even stops replication of the virus, then you can't infect the next person. And we know that this is possible to do. This is what we do in HIV. We're very interested in this in SARS as well. And it's possible that a vaccine would work by, again, letting you get infected, but reducing the concentration of virus in your nose. Active immunity is vaccination, where we can see individual level benefit or a population level benefit or both. And then if we infuse antibodies we made in a lab into you, we call either through convalescent plasma, that is taking blood from somebody who's recovered and giving you their blood, essentially their plasma, or we can make hyperimmune globulin. We're doing that in cows and in humans now, but humans we harvest it, cows we can make it in, and then monoclonal antibodies have attracted a lot of attention. So vaccines first. a strategic plan. My colleague, my partner, Dr. Corey, along with others, published the strategic plan for vaccine development, and this is the bottom line. You see on this slide, we have 3 kinds of vaccines. The newest one, which is a tour de force of technology that's waited a long time to realize its potential, is mRNA, where the RNA is put into a lipid and given to you, and your cell makes the vaccine for us. And that's what we've done in the United States. We can make a dead virus. We can package what's needed for the immune response into a virus, call it an adenovirus, and kill the adenovirus, give you a dead virus and inspire an immune response. Or most vaccines you've taken to date are proteins with a thing called an adjuvant that makes it more immunogenic and makes your arm hurt, and we can do all 3 platforms of vaccines. These are all our vaccines now. Moderna and Pfizer are mRNA vaccines, and that's what we're using in the United States because it requires special conditions to store. AstraZeneca and Janssen made adenovirus vectored vaccines, and they're being used, and Janssen made a 1-dose vaccine. They don't look quite as good as the mRNA vaccines right now, but we don't really know because time has not gone by. And they've been used, AstraZeneca has been used a lot in Europe. It recently failed with the South African variant, so it's out of Africa. The Janssen vaccine works partially in Africa, so Janssen's still alive in Africa. And then we have 2 other vaccines we're just finishing trials of, including on this campus, one a protein called Novavax and the other a protein called Sanofi. But so far Sanofi is kind of sucked, so they're way behind. And this is all the vaccines. It is our intention to vaccinate at least 100 million Americans in the next 3 months, but that requires more than 1 million vaccines a day. These companies have lots of money to gear up, so we're very hopeful we'll get 100 million people vaccinated. And as you might guess, we're trying to vaccinate the people at greatest risk for progression disease. The problem is those same people may not respond as well to a vaccine as a younger person, so we just don't know this yet. The other problem is we don't really know what this vaccine is doing exactly. So far it's proved safe, but the endpoint that was pursued for the vaccine has been, did we prevent you from progressing your disease? We call this V disease. And most of these vaccines prevent people from getting in the hospital. They prevent you from dying. But we don't know whether they prevent you from getting infected. We call that V susceptibility. And we don't know if you get infected, whether we prevented you from infecting the next person, from your nose to their nose. We call this V infectiousness. So it is likely in the next 3 weeks, we'll start a study on this campus that will recruit about 600 students here. And we'll start a study on 20 campuses to try and answer these questions, where college students will be given they'll be divided into an immediate vaccine or a delayed vaccine. Everybody gets vaccinated. And if they swab their nose every day, we can work out all these questions. So we probably will start this. It looks very good. We'll start this study at 20 universities in the next few weeks. And again, this is what was measured in these studies. They simply said to people, did you get sick once you got the vaccine? And for most of these vaccines, far fewer people got sick that got the vaccine than if they didn't get the vaccine. And I think this is, there's a slide that's not showing, interesting. Well, if you could imagine, why is that slide? It should be here, it's not there. If you can imagine, this is the Pfizer vaccine. Why it doesn't, you guys are smart, why do I not see this? You don't see anything either, right? Somebody unmute themselves.
We don't see it.
I don't see it right now.
If you saw it, we'll just show you that with the Pfizer vaccine, for example, when you compare the experimental group, the 30,000 people are enrolled. So 15,000 got a placebo and 15,000 got the vaccine. And when you look after 7 days, and there were 2 doses, but after 7 days, there's a huge difference between the vaccine group and the placebo group. So the placebo group keeps getting infected. The vaccine group stops getting infected. And then by the end of the study, at day 20 something or whatever it was, that's probably day 57 maybe. By the end of it, there was a 95% reduction in people getting sick from COVID No one died from COVID. No one got hospitalized from COVID. So that was considered a big home run. So V disease was affected enormously by Moderna and Pfizer. For Janssen, the adenovirus, it was about 72%, but a different population. And we don't have the results of the proteins yet. So that's our vaccine program is really important. And if the vaccines actually work beyond V disease and stop V infectiousness, we ought to see a dramatic effect on new cases. Now, the Israelis are trying to vaccinate the whole country in a month or two. And they already argue that in a program in Israel where they vaccinated a few 100,000 people, that they're seeing a 30% reduction in new cases in the population that surrounds the vaccinated population. That's extremely encouraging. We would love the vaccine to stop transmission of COVID. Now, there's alternatives. People who get COVID have an immune response. We can bleed them and take their plasma, and that's called convalescent plasma. And that was approved by, first the FDA said they're not going to do it, then they did it, and they let us bleed a person and give somebody else's plasma to somebody else. It really doesn't work that well. It really only works if you give it early, and it really only works if you use plasma that has a lot of antibody called high titered plasma. Turns out that not everybody, I already told you this, not everybody makes high titered plasma. An asymptomatic person is not going to make high titered plasma. So convalescent plasma, we have a program here. We collected a lot of plasma. We've titered it. We only use the high titered plasma. But there's a better approach, really. Convalescent plasma is limited because it's difficult to obtain. If we make the epidemic go away, there'll be no more plasma. We don't really know everything plasma does, and so on and so forth. So the alternative that we've been working on are these things called monoclonal antibodies. They offer immediate protection. So if you say you have an underlying disease and you think you're going to die from COVID, if we infuse a monoclonal antibody in you, whatever benefit you're going to get right then. They can be provided to people who might not respond well to vaccines, because it turns out that a lot of older people probably won't respond well to vaccines. That is, they won't get a good immune response. They could be used for treatment and stop viral replication and block progression. They can help us to understand vaccines. So who should get a monoclonal antibody? People living in nursing homes if they're not vaccinated, because those people are at grave risk of dying. And I'll share with you that I led a study that's being published in the next few weeks where we showed if we infuse a single monoclonal antibody called Imdevimab to people in nursing homes, we reduce the cases of COVID by 80% and we eliminate death. So that's a pretty big home run for a single monoclonal antibody. We could also use these in high incidence workplaces, and we can treat people that are going home to their household and are infected, hoping that we're going to stop the replication of the virus and stop them infecting other people in their household. We could also give people in the household these monoclonal antibodies to protect them from the index case, the infected person. So this slide you may not like, but I had to show it to you. This is how technology is really important. This is a slide of how we can so quickly make a monoclonal antibody. We just have to bleed a person who's had COVID. And then we bleed that person and we look for cells that make antibodies against COVID. And we look for a super duper antibody made by a super duper cell. And if the cell is then taken out of them and it can grow outside their body and replicate, we can put it into giant vats and make as much monoclonal antibody as we want. We then take the monoclonal antibody, we put it in a small animal, usually a hamster or a mouse, and we show that when we give the hamster or the mouse the antibody, either for prevention before infection or as treatment, the animal is happy. We say, whoa, let's do a human experiment. Now, COVID first appeared in the United States in the middle of January, and by the beginning of February, many companies had already done all this and made monoclonal antibodies ready for human trials starting in March or April. So this is a technology that has taken many, many years to develop, but is an amazing technology. Just like the mRNA technology is amazing, because we can swap out antibodies very quickly. If a virus became resistant in the United States, we can say, well, we're no longer going to use antibody X, we're now going to use antibody Y, and we can make new antibody. And the FDA has been very permissive about swapping out antibodies so far and combining antibodies. there's not just one or two antibodies. These are just a few of the companies that are making monoclonal antibodies because the technology is so robust. We've worked a lot with Lilly and Regeneron. They're farthest along. Lilly has approval already for a combination of antibodies for treatment for early infection, as do Regeneron and Lilly. Both companies have approval for treatment. And last week, both companies asked for permission to use similar combinations to prevent infection in people at high risk. This is called emergency use authorization. And that's going to be followed then by the request for what's called the BLA, a license. And these antibodies do work, and many more antibodies from this and other companies are in development. Some of these antibodies can be made differently. Like Vir has made an antibody, as has AstraZeneca, where they modified it so it'll last 6 months. AstraZeneca claims their antibody might last a year. So you can see if you're not going to use a vaccine, we might be able to use antibodies for prevention as an alternative to vaccines if they're going to last that long. So these monoclonal antibodies are a big deal in the COVID space. It takes us, if we were going to use like a small molecule, like the antibiotics you got when you get sick, like penicillin, it takes us a lot longer to make penicillin than it does to bleed somebody who's recovered from an infection and make a monoclonal antibody the way we've done with COVID. Lilly's drug, I told you, was approved, and just yesterday, their new dosage forms, their combinations were approved. Regeneron's been approved. Both are going for prophylaxis. I guess this is just a reiteration of the situation with the spectrum of disease. And I guess I put this in here to point out that the 3 legs of our stool, behavior change, vaccination, monoclonal antibodies for prevention, and then early treatment. And early treatment could be with an antiviral therapy. Scientists at UNC have made an antiviral therapy that we're testing with Merck, or antibody therapy that's already been approved. That's to prevent you from getting in the hospital. Once you get in the hospital, the virus isn't your problem. It's the inflammation is your problem. And we have a whole different cookbook of dealing with inflammation and blood clots and other stuff that happens in the hospital. Knock on wood, none of you will ever get that sick with COVID. The Lilly antibody study that allowed them to get their EUA is called BLAZE-1. And that BLAZE-1 study has not been published yet. It shows a 70% reduction in people getting sick. So you get COVID, but you don't get sick. It showed all the deaths that occurred in that study were in the placebo group. No deaths once you got their antibody. Regeneron makes drugs called Casirivimab and Imdevimab, very similar results. And on this slide, though, I don't know that I show it to you. I want to point out to you that we studied the nose. And this slide, if you look at the left-hand panel, and you look at placebo versus Regeneron antibodies, and you look at people who have not yet made their own antibodies, you can see a pretty large difference in the concentration of virus in the nose. And it's wiping the virus out of the nose that saves the person from getting sicker. With our Lilly study in old people, these are young people, and even their noses are benefiting. But with old people, we showed a huge difference in the nose between the placebo and the experimental bamlanivimab. Now, for people who are already making antibodies, which would be almost all the young people are going to make antibodies very quickly, we're not going to be able to see much benefit from treating them because they're going to deal with the infection with their own antibodies, as shown on the right, very, very quickly. I told you we're making our own small molecule. We have worked with other companies on a small molecule called EIDD2801. This is a pill. You take it once a day for 5 days. Merck took over the studies and is doing a really large study to see whether this drug will wipe out the virus in the nose and stop progression of disease. And in the end of the day, this is what's going to happen. So what's going to happen in 6 months or a year from now? Either we're going to have to keep using masks or masks are going to go away. That's going to depend a lot on how well the vaccine actually works. And COVID is eventually going to go from epidemic to endemic. That means we're not going to have these giant clusters. We're going to have occasional cases. At a college, for young people, a case of COVID would probably just be, you have to go home and be isolated for a week or 10 days. For somebody at risk, we're going to put them in this CVS doc in the box and give them something. And that something might be a monoclonal antibody or might be something else. And the purpose of this will be to prevent them from requiring hospitalization or going on to severe illness or death. This doc in the box exists already at most of the CVSs in North Carolina. If you go on 15501, you'll see this sitting there. I don't think it's really been used yet, but it will be used because we can't safely take people with COVID can't easily go to a private doctor's office. And we're not going to forever be able to manage all the people with COVID endemically without the help of pharmacies like this. Lastly, I'll point out to you that we do lots of COVID studies on this campus, and I've already warned you, if things go right in the next 3 weeks, there'll be an advertisement that'll say we need 600 college students to be vaccinated. So you would get out of line and get a vaccine. And the way it's going to work is half of you would be vaccinated immediately and half of you would be vaccinated in July. The way you can be eligible for the studies, you have to be on campus for the next 5 or 6 months. And you have to agree to be paid to swab your nose, not painful swab, just a simple swab, every day for 6 months. It's a pretty easy study, and you get $10 a swab. So if you see this advertised and somebody's interested, they should sign up. We think we can enroll 500 students in 2 or 3 days. So I'm trying to make you aware of it. I think that's my last slide. We have about 15 or 20 minutes for questions. I'm going to stop sharing my screen. Look at myself. That's what we know about COVID.
I have a question, actually. You said that the one that you were leading on UNC's campus is a protein with adjuvant. What made you decide to go in?
If we get this off, well, okay, good. What studies have we done on this campus? We have done 2 studies on this campus already. So we have a vaccine research site next to Harris Teeter on 86, next to a pizza parlor. So it's where it used to be where they had like some Tar Heel museum or something. It's out, out on 86. There we have examining rooms and a clinic. At that site, we did the Moderna vaccine, and that study's done. Now, this week we're completing, and that is a mRNA vaccine, and the study's over, and the drug is under EUA already. We're completing the Novavax study this week. 30,000 people have been enrolled. I think we're probably, if we're still taking volunteers, it's the last few days. If you want to volunteer, what you do is go on that website, which I showed you, go to UNC COVID or COVID Prevention Network website, and then you just sign up. And if we're still taking volunteers, you can volunteer for that study, the Novavax study. We anticipate vaccine being available for college students before the fall semester. So it's not like we're going to go forever and not offer you vaccine. It's a big debate now, depending on what happens next, whether we'll mandate. Some vaccines are mandatory for you. You can't get on this campus if you haven't had a measles vaccine, for example, or had measles. So whether we mandate COVID vaccine depends on what we learn about the vaccine. How safe is it? How powerful is it? Is it critical to the well-being of your fellow students? And what's the relationship with masks? That's a decision that no one's touched. But we're learning a lot about vaccines very, very quickly. Did I answer your question?
Yeah. The other question I had was, why?
Sign me up. Amber, you could be our first. Okay, so Amber, you could be our first volunteer. Can you remember a name if I tell you this?
A name? Okay.
A name.
Okay.
Why don't you email? You can email Dr. and you'd have to go and address Sylvia Becker-Dreps. It's a pretty easy name.
Okay.
She's going to run the study on campus and say, I was given a lecture, and I pointed out we're doing the study, and if she gets the study off the ground, sign you up. And then she'll have your name, and you could be the first volunteer. You could be the poster girl.
Oh, I love that. Okay, only if
Sylvia Becker-Dreps. Send her an email right now. We have a budget already. We're just waiting to see. We need 24,000 doses of the vaccine.
Mhm.
We need a lot of money. more money than you could believe to do this.
I can only imagine. But
But
when
I believe it's going to happen. And my colleague, Dr. Corey, and I are in charge of this at some level. And then Audrey Pettifor, Professor Pettifor, she and another physician from Harvard are running it. And every campus of 20 campuses have a person like Sylvia Becker-Dreps. But I am sure she would love you to be the first person signed up.
Sounds good. Sounds good. And I'm gonna have.
Tell her you're excited about it.
I am excited about it. I'm already volunteering at COVID testing sites, so I already, I'll probably be in a position to swab myself most days of the week. So that sounds good.
Well, 2 thumbs up.
Sounds good. But thank you so much.
All of you can volunteer. By the way, if you want as a group, if you want to explain to Sylvia, you all belong to the Carolina Cancer Association, you all want to volunteer, that would be even funnier. And then we would say the entire Carolina Cancer Association volunteered for what we call the college vaccine study.
I see. No, this could be a way for you guys to get active membership. How do you feel?
That would be awesome. That would be awesome, awesome, awesome.
Yes, Kylie, let's do it. Okay. Well,
I'm ready.
Of course, none of this is ready yet, but
okay.
It's aspirational, but it might happen. What's your dog's name?
Oh, that was Bandit. That was Bandit. And excuse Gabby and I for being in the same screen. We're both part of CCA, so we were like, oh, we might as well just join together instead of pretend like we're in different rooms. But dude's morning.
I think it's very difficult. Let me say one thing. The university, as a university administrator, we're very sensitive how difficult this has been for the students. It's difficult for old guys like me, and it's difficult for the students. So hopefully we can create this semester. Knock on wood, has gone great so far. We have a model, and you're being tested once or twice a week, and the percentage of testing is below what we anticipated. So we're very excited. We'll see what happens with the Duke game, but we're very excited about how well you're doing. We know how hard it is. So I do feel there's this sense sometimes the students feel like, especially the Daily Tar Heel portrays that like, the administration is the enemy of the student. Everything we do is bad, wrong, mean-spirited. That really isn't the case. We're thrilled that the students have a good experience, and we're all a third way through the semester. 2/3 more, and we'll have succeeded. Other questions, gentlemen?
I know Sloan had another one, so I'll let him go first.
Okay. Yeah. Do you? You told us there were 3 main types of vaccines. Do you know what kind of steered certain companies to try out one or the other?
Yeah, that's a really good question. So why did one company, Merck dropped out. Sanofi had to slow down. So each company did what they thought they could do well. And in the case of Moderna, that was like a worthless company, but had been working forever on mRNA vaccines. So when COVID came along, the speed with which they could make the vaccine became a huge advantage. That's the first point. For Pfizer, giant company, they were working with a company called N-Biotech, a tiny company in Germany, and they had the same thought as Moderna. We know how to do this. Let's just do it. They didn't know it was going to be safe, and they didn't know they could make an immune response. So none of this was known. Big risk, right? So those 2 companies went with technology, and their technologies are only appropriate to rich countries, okay? Because you need big freezers and a cold chain and a lot of other stuff. The second 2 companies with adenovirus, one of them, Johnson & Johnson, Janssen, they had made an Ebola vaccine for adenovirus successfully. That is the Ebola virus vaccine. So if we were sending you to Liberia, you would take an Ad26 vaccine. So they had demonstrated safety already. So it was easy for them to say, and they had a collaborator named Dan Baruch, who's another one of our partners, and he was able to help them engineer a vaccine. And AstraZeneca, they didn't make a vaccine. They started working with the people at Oxford who for years have been trying to make a vaccine for HIV using a chimp adenovirus, not a human adenovirus. The reason they didn't want to use a human adenovirus is because then in the second shot, you might make an antibody against the adenovirus, then you won't get a benefit. And then the third company, Novavax, that was always a protein vaccine company, a tiny one. It became bigger, and Sanofi makes lots of vaccines. Ironically, Sanofi's vaccine caused people's arms to fall off. It was like they put the vaccine in people's arms and they turned so bright red and their immune response was so terrible they had to slow down. So the company that you'd think would be the fastest turned out to be the slowest. But all of these, and there's not a lot of other vaccine companies. Those platforms are all the platforms. And probably the students, for the college study that Amber is leading, she's going to get the Moderna vaccine for sure, okay? But in the summer, probably the students will get the Janssen vaccine, the adenovirus vaccine. The reason why is because it may not be as effective, but you don't need a vaccine as effective as a nursing home person. And we still don't know about V susceptibility and V infectiousness. And those are really big metrics. Okay, other questions?
In terms of the study, so obviously we'd have to stay on campus throughout the whole duration, like for the next 6 months.
No, you don't have to be on campus. You just have to be able to swab your nose every day, and you have to be in somewhere near enough, but you don't have to be a student enrolled.
Okay.
I don't know where your home is, Amber, but let's say are you from North Carolina?
I'm actually from Virginia, but I'm in North Carolina right now.
Well, but how far in Virginia? Deep in Virginia, or?
I'm in Northern Virginia.
Yeah,
So
I mean, I think it just depends. We need to enroll students who, if they develop symptomatic illness, the swab comes every day, but we need to be able to see you if you don't feel well. So that's why we need you on campus,
Okay.
or near campus. So students who live in Chapel Hill, Hillsborough, whatever, they're great candidates.
Gotcha. Well, that's
Probably
interesting.
we could pay the entire CCA dues for all of CCA.
There we go.
I could probably take over the dues. How much are the dues for a year who belong to CCA?
Just $5 right now. Yeah, that's
Okay, I personally,
half a day.
if all the CCA join this study as a group, I personally will pay the dues for a year. No problem.
Ooh, I love these incentives. It's looking good.
I'm giving you a lot of incentive. Tell Amber, when you talk to Sylvia, tell her I promise to pay all the CCA, Joe, but not everybody's gonna be around for both semesters. We need students who are around. You can't be.
I'm gonna hold you up to that.
Yeah, that's a limitation. Poor Amber is lower on the list now.
Oh
Okay.
no, I'm staying throughout the.
If you're staying, you're in.
Yes, I'm from Virginia, but I'll be on campus for quite some time.
Okay,
so.
then you're the number one. You're going to be student.
I can't wait.
Subject one.
I'm going to be disappointed if I'm not the poster child for
You will
this.
be the poster. We will make a poster of you raising your fist and.
Oh gosh. So I did.
Email Sylvia tonight. Tell her how excited you are.
I was just telling Gabby I'm drafting the email as we talk. Well, I don't want to.
She'll know. I told her I had to give a lecture. So you're giving a lecture, and if the study gets off the ground, you want to be subject one.
Awesome. I better. This is gonna look. This is bragging rights.
You'll make if you get $10 a day roughly. I think that's what we're planning on paying.
That's a lot of money.
That's a lot. You get $70 a week for 6 months. You get $500, $600, $700, $1,000. I don't know. They could get cheaper. They could if they see too many students interested, they might make it $750. It could, but either way, you'll be altruistic, and I'll pay your CCA dues.
Okay, that's
wow.
All right, let's keep going.
I did have a question with regards, because you mentioned the Duke game, so I was just wondering what, like from your perspective, like kind of went into that decision to still have on-campus classes starting on this past Monday.
Yeah.
With that,
I think okay. So all along, so let me have it by way of disclaimer. Like I'm a university administrator, right? But I play a lot of roles at the university and advise them about what to do. There's and COVID has a biological metric and a public relations metric, and they're not the same. The classrooms that you would go to for in-person classes, we have maybe 20% in-person classes. With masks and all that they did for those classrooms, there's almost no way to get infected in a classroom. And the faculty, it's very hard to convince them that you don't represent this huge danger to them because you're 20 years old. You're wearing a mask. Some of you are very responsible. They're standing in the front of the class with plexiglass. They're wearing a mask. They're never going to get infected. And in fact, many studies done at other universities have shown the classroom is incredibly safe. So that's the first point. So having classes in person has almost nothing to do with the Duke game. In other words, that decision, whether you're going to have classes in person or not in person, would be really a public relations decision, not a biological decision. If we thought it was dangerous to be in that class for you or a faculty, we would have no in-person classes. That's the first point. The second point is that the real jeopardy of COVID lies in the housing and the social intercourse. It lies in, if you're in a room, if you live in a house where people are casual about their relationships, and if one person gets COVID, they can infect the entire house. Fraternity houses are now considered a house. So if one person gets COVID, everybody has to be quarantined for 10 days or 14 days. It's like a household. For dorms, we try to reduce the occupancy. The sampling of the nose also gives us a lot more control over what to do if you get infected. Now, if you get infected, you're either getting infected in your room or in a restaurant or in a bar or at a party with your friends, but not in a classroom. But that's biology versus public relations, and being sensitive to how afraid people are. Infectious diseases are scary. Have any of you seen Ben-Hur, the movie? Remember the lepers living in the cave? That's like every child's nightmare is, infectious diseases are very, very scary, including COVID. They have a lot of stigma associated with them. But on the other hand, I think the chancellor's decision to go forward with classes kind of had everything to do with the idea that we're sampling very frequently and that the classrooms have been rendered safe. Now, next week, we're very curious about what happened on Franklin Street. So next week, when we get all your samples, some of you get sampled once a week, some of you get sampled twice a week. When we get all your samples, either we're going to see a giant spread of COVID because we beat Duke or not. And I have no idea. A lot of the kids on Franklin Street were wearing masks. It wasn't like everybody was without a mask. You were outside. There's a lot of reasons we might escape a disaster, or we might see a lot more cases, which we don't want to do. But if we do, we'll deal with it. We're not sending you home no matter what.
So from an epidemiological standpoint, moving forward, what actions do you anticipate the university making that can sort of keep up our performance with reducing the spread of COVID?
Well, I think the university is trying to be stern but judicious about behavior. They realize you're 20 years old or 21 years old, okay? And they're trying to say, look, we don't want you to throw you out of this university forever, but we don't want you, disobeying the kind of rules that have been set, especially for students living on campus. And for those living off campus, there's a set of rules and behaviors expected. The local police and the town police are much more severe now about gatherings, right? So the gatherings have been really a source of kind of, I guess, honor code violations or whatever you want to call them, where students who are in gatherings, it's like, why did you do that? We asked you so much not to get COVID so we can get through a semester. I know there's been a problem that's been a source of great consternation in the university about an Instagram issue. You guys know about this? About kind of citizens arrest. And of course, we don't want any students feeling that they're responsible for another student, right? I mean, that idea of whatever students are involved in feeling that they feel compelled to make a citizen's arrest, it's probably not a good idea generally. We'd like students to be part of a team, let's get through the semester. Things will look different in the fall. All of you want, all of you want an education, I know that, and all of you want to have an experience that's like reasonable. It's pretty hard with a mask, and it's hard to eat, and socialize. So it's a rock and a hard place. So, okay, so the university. So the first thing is get through the semester. Understand whether all the testing we put in place is helping us. The modeling we're doing, is it helping us? Second, let's see what happens with vaccines. How well do they work? If we have vaccines for students, is that going to drastically change the campus in the fall? And if we have vaccines for students, how do we inspire students? We are huge leaders in influenza. Like UNC is the most influenza vaccinated university in the country. So I would have every reason to believe if we had a vaccine to give you for COVID that works and is safe and that your parents agree with, that you would probably be thrilled to take it, because you don't want to get COVID. So So we'll see. On the other hand, we're well aware that the students feel invulnerable, partly because they're young and partly because of the slide I showed you. You're the poster children for not getting sick. So it's pretty hard for me to give you two messages. Wear your mask all the time, like Sloan is. And by the way, even if you get COVID, I'm the one who's going to die, not you. So it's a mixed message, and it's tough to give mixed messages. Sorry for the brutal honesty. Did that answer your question?
I'd say so.
Okay, other questions? You already spent the CCA hour and the best CCA lecture ever. You all ought to decide, instead of the Carolina Cancer Association, you want to change yourself to the Carolina Infectious Disease Association, CIDA, and tell the Cancer Center people that they're boring and you've decided to move on into something more dynamic and more interesting. And mention that I thought you should change your name. If you have any relationship, say, it was my idea you should abandon cancer and move to infectious disease, which would greatly amuse.
I'll tell Dr. Baldwin about your suggestion.
Yeah, please.
Yeah.
Please.
Yeah.
Tell him exactly that. I said, I gave a lecture to you about, is he your sponsor, Al Baldwin?
He is. Yeah, Al Baldwin.
Please say, look, the guy was giving a lecture and he said, why don't you just abandon this PCA thing and we'll embrace you as an infectious disease group. That would be a good laugh.
I'll let him know. Yeah, he would enjoy that.
All right. Anything else, ladies and gentlemen? All right. Thank you so much for inviting me.
Thank you so much. It was
Thank
a great
you.
lecture.
Thank you for coming.
you.
Yeah.
That was great.